So let's look at nutrient exchange in mammals. So we've got 3 syllabus objectives, and the reason for that is that I've grouped these first two together because I think that the exchange surfaces, the properties, structural features of these exchange surfaces in the digestive system and the respiratory system share so many similarities, it makes sense to do them together. So we're looking at the digestive and circulatory systems of mammals and how we get nutrient exchange, and also the respiratory and circulatory system of mammals and how we get exchange of how we get gas exchange there as well. As well as that, we're going to look at how a closed circulatory system facilitates this efficient transport of material. So what nutrients are there that the body needs? Well, we need oxygen and glucose for cellular respiration. The oxygen obviously comes in through the lungs. The exchange surface is called the alveolus. And of course, that's also the surface that's involved in the removal of carbon dioxide waste. Now, we also need amino acids and fatty acids as well for the synthesis of macromolecules in the body. And of course, they are also, the exchange surface for them is also in the small intestine at the villus. So let's firstly look at the alveolus. So it's actually really good doing both of these together because the nutrient exchange surfaces in both the alveolus and the villus, they both have the same characteristics. Firstly, they have a large surface area. So the actual gas exchange, or the functional unit of the lung for gas exchange, is the alveolus. And alveoli is the plural, alveolus is the singular. So you can see that they're like grapes, they're these air sacs. This is like a cross section of what one would look like, and then as you can see, there's this is what it looks like when you've got like a bunch of grapes, if you like. And I want you to understand that all of this pink stuff here on the lung, well, they're all the alveoli. So it's got a really, really large surface area. So much so that the inside surface of the alveoli, if you spread them out, they would be about the size of a tennis court. So it's really quite large. So we have a large surface area as the first characteristic. The second is the alveolus wall is very thin. It's one cell layer thick. And so that allows that there's a really short diffusion pathway. It's wrapped in a capillary network. You can see here's a single capillary here, and you can see a capillary network wrapped around the alveoli. And also there's the surface is moist. So whilst it's carbon dioxide and oxygen gas, it's actually dissolved in water at the exchange surface. And the last thing it's about, there needs to be a concentration gradient. We maintain this concentration gradient through breathing in and out, breathing in oxygen and expelling carbon dioxide. That maintains low carbon dioxide in the alveolus and high oxygen. And we've also got perfusion, which is the movement of blood through the capillary network that brings high carbon dioxide and low oxygen blood back from the body. So in the capillary, we've got a high concentration of carbon dioxide and a low concentration of oxygen, such that the carbon dioxide is going to go down this concentration gradient into the alveolus, and the oxygen is going to go across this concentration gradient from the high concentration area of the alveolus into the low oxygen area of the capillary. And then if you have a look at the nutrient exchange features of the small intestine, again we have a large surface area. Now there's 3 reasons for that. First of all, the small intestine is very, very long. So that gives you a nice long surface area, large surface area just by the pure length. But we also have these finger-like projections called villi in the lumen of the small intestine, and that increases surface area significantly as well. But as well as that, the cells that lie in the villus actually have finger-like projections of their own called microvilli, which again increases surface area further. Again, we've got a thin, one single cell layer thick wall of the small intestine. So again, there's a short diffusion path. We have a capillary network, but this time the capillary network is on the inside of the villus. We have, again, it's a moist surface, and again, we've got a concentration gradient. The reason for that is we've got a higher concentration of our nutrients in the small intestine than in the blood. And of course, the blood is continually pumping once the nutrients move into the from the small intestine into the capillaries. It's going to continue to pump that away and bring in blood with low concentrations. So the first syllabus objective was about the structural features of these exchange surfaces. So just to summarize, there's 5 aspects: large surface area, thin walls, so allowing for a short diffusion path, concentration gradient, moist surface, and a large capillary network. That's our 5. The next thing we have to look at is to describe how a closed circulatory system facilitates the efficient transport of materials. Let's see what we mean by a closed circulatory system. Look, essentially, a mammal has 2 circulatory systems. We've got the system called the pulmonary circulation, which takes blood from the heart to the lungs and back again. So that's that process of oxygenating the blood. And we've talked about the alveolus with the capillary network around it. But it's a closed system because the capillaries don't go to dead ends. We go from arteries to arterioles to capillaries, and then it continues on as capillaries that head back to the heart, capillaries, then venules, then veins, and so it's a continuous closed circuit, and it's the same thing around the body as well. So that the systemic circulation takes blood from the heart around the body and back to the heart. So it's taking blood to the small intestine, but it's taking blood to the muscles and the heart, etc. And again, it's a closed system going from the arteries to the arterioles to the capillaries, and then from the capillaries to the venules to the veins. So it's a closed system. So again, let's have a look at the characteristics of these capillary beds. Because they're wrapped around these exchange surfaces, and they've got very thin walls. The capillaries are very small with very thin walls, and they're wrapped around these exchange surfaces. So again, we've got a short nutrient exchange path, and because we've got this whole network of capillaries, we've got a large surface area in which the nutrients are able to diffuse across into the capillaries. And the capillaries form a closed, and it's a closed network, as we said. So from the arterioles to the capillaries, and then to the capillaries back to the venules. So it provides an efficient nutrient exchange because we have this large surface area with these thin walls, and of course the capillaries are very, very close to the nutrient exchange surfaces.